Today we will discuss spermatogenesis. What is spermatogenesis? Spermatogenesis is production of male gametes, that is sperm, in testes. So it is production of sperm. And where this production takes place? It takes place in testes. Testis is singular, testes is plural. So if we see the humans testis in section, in transverse section, then we will find that there is a tough capsule known as tunica albuginea. It is tunica albuginea, in which septa are there, and in between the septa are the highly coiled tubules, which are known as seminiferous tubules. These are seminiferous tubules. Seminic means semen, which means seed. Ferrous, that bears, that contains. So semen or seed bearing tubules. These are highly coiled and very long tubules in which spermatogenesis takes place. All these seminiferous tubules join to form a network known as testes. This network is testes, in which high amount of estrogen is there, and here concentration of sperm takes place. This leads into epididymis. From that epididymis arises the Wells differential. This is head of epididymis, and this is tail of epididymis. So in this epididymis also tubule is there. So production of sperm takes place in these tubules, these highly coiled tubules, which are known as seminiferous tubules. If we see the section, the transverse section of this seminiferous tubule, that section will reveal that there are present the different developing stages of sperm. Also, there are present other cells known as interstitial cells of Leydig or Leydig cells, which produce androgens, testosterone, DHE, etc. That testosterone is converted into estrogens in retitestes. So now we will see the transverse section of a seminiferous tubule. A seminiferous tubule has a basal lamina. The outermost layer of the filament is basal lamina. Every epithelial layer has a basal lamina that provides support to the epithelial layer. Inside this basal lamina are present the Sertoli cells. Sertoli cells are the modified epithelial cells of seminiferous tubules. These are Sertoli cells. These Sertoli cells make tight junctions. These form tight junctions, and in these ridges, in these clefts in between the Sertoli cells are present the various developing stages of sperm. These tight junctions form blood testis barrier, and that barrier prevents autoimmune attack on developing stages of spermatogonia or sperm. So, Sertoli cells not only provide protection to these cells, these also produce androgen binding protein, which is necessary for spermiogenesis and spermatogenesis. Androgen binding protein. Androgen binding protein, what it does? It binds with testosterone and maintains a high concentration near the developing sperm. Androgen binding protein, which is produced by Sertoli cells. And what is the source of this testosterone? The source of testosterone is interstitial cells of Leydig, the Leydig cells. These produce testosterone. And here androgen binding protein is there. That protein will bind with testosterone, and that will maintain a high level of testosterone near the developing sperm. In the center there is lumen of seminiferous tubule, in which the mature sperm are released, the phenomenon known as permeation. So now we will discuss step by step the formation of sperm. So there are three phases of spermatogenesis. The first phase is proliferative phase, the second phase is meiotic phase, and the third phase is spermiogenesis. The first phase of spermatogenesis is proliferative phase. Second phase is meiotic phase, and the third phase is spermiogenesis. So we will discuss one by one. First is proliferative phase. Proliferative phase, in which the stem cell proliferates, that divides by mitosis. It divides by mitotic division. The primordial germ cells during the early embryo stage, in the embryo, the primordial germ cells, these arrive at the sex folds to become gonocytes. sex codes will form seminiferous tubules. We are talking about the developing stage, the embryo. Within the embryo, these primordial germ cells arrive near sex cord. They incorporate into sex cords and become gonocytes, and these gonocytes then become spermatogonia type A. These gonocytes are stem cells. These are true stem cells. If these are injected in an adult male testis, then these start to multiply at a very high rate with the mitotic division. So these become spermatogonia type A stem cells. So these keep on dividing, and at maturation, at puberty, when the spermatogenesis is resumed, the spermatogonia, these become type A1, then type A2, type A3 and type A4 by multiple divisions, by mitotic divisions. But each time they remain attached with the help of cytoplasmic bridge. So these become syncytium. Syn मतलब साथ together, cytium cyto cell. बहुत सारे cells एक साथ, that is syncytium, जिनका cytoplasm fused हो. So these now become type B spermatogonia, and which travel through an intermediate stage. Transition state is intermediate spermatogonia. Type A4 spermatogonia will lead to intermediate spermatogonia, and then these will become type B spermatogonia. So this is proliferative phase. Up to this stage, formation of type B spermatogonia, it is proliferative. Now we will discuss the second phase of spermatogenesis, and the second phase of spermatogenesis is meiotic phase. In meiotic phase, the type B spermatogonia will divide once by mitosis to become primary spermatocytes. These primary spermatocytes will enter the meiotic phase. These will undergo meiosis, the first meiotic division, and these will become secondary spermatocytes, and these are still attached to each other, and so many cells. There is a series of cells which remain attached to each other with the help of cytoplasmic bridges. So now the stage is secondary spermatocytes, which will undergo second meiotic division, the meiosis, and again these will remain attached, and these will become spermatocytes. These will become spermatocytes. This is the meiotic phase. Then the developing sperm will enter in third phase, that is spermiogenesis, that we will discuss now. The third phase, the spermiogenesis. All the cells which have formed up to formation of spermatids do not look like a mature sperm. These are only cells which are joined together in a series with the help of cytoplasmic bridges. Nearly 512 cells are formed from single spermatogonium. So 512 cells remain attached in a series with the help of these cytoplasmic bridges. What is the significance of these bridges? It might be ensuring the equal survival of each cell, whether it is containing X chromosome or it is containing Y chromosome. So we have seen that spermatids have been formed during meiotic phase, which are haploid cells. Now these will undergo spermiogenesis to become mature sperm. So what are the changes which take place during spermiogenesis? Those changes are as follows. These cells discard excessive of their cytoplasm. They generate a flagellum. A flagellum is formed. Excessive cytoplasm is discarded. Acrosome is formed. Acrosome is a cap-like vesicle which is present on the haploid nucleus, and within this nucleus the genes will not express. These do not express. At this stage, in a mature sperm, no expression of genes takes place. So, in order to ensure that thing, the histamines are replaced by glutamines. Glutamines are formed, which are histone-like proteins. These cause compact binding of DNA. Compaction of DNA takes place. Flagellum rises, excessive cytoplasm is discarded, and these are released in the lumen of seminiferous tubules in the form of mature sperm, and that phenomenon, the release of sperm in the lumen of seminiferous tubules, is known as spermiation. So we have discussed the three phases of spermatogenesis: the proliferative phase, meiotic phase, and spermiogenesis. In case of proliferative phase, mitotic division takes place in the spermatogonia. In the meiotic phase, meiosis takes place, during which primary spermatocytes become secondary spermatocytes, and then secondary spermatocytes become spermatids. And during the third phase, that is spermiogenesis, the spermatids discard excessive of cytoplasm. They generate a flagellum. They condense their nucleus. A vesicle known as acrosome is formed, which contains lysosomal enzymes, protein degrading enzymes, hyaluronidase, etc. And also the protamines replace histone proteins. No expression of gene will now take place in this cell, the mature sperm, and these mature sperm will be released in the lumen of seminiferous tubules, and that process is known as spermiation.